I wanted to go over the blood type problem with you here. I'll show you the answers for this page, which is going to be page 98 in your notebook. But I wanted to talk you through the baby question. When you see a question like this on your assessment, because there will be something very similar to this on your unit test, I want you to make sure that you write out some of the details, because this kind of problem is too easy to uh, get tangled if you do it all in your head. So it says we've got 3 babies that were in a hospital and they got mixed up. We know the parents' blood types, but we don't know if they're homozygous or heterozygous for certain blood types. But every group of parents or pair of parents is going home with a baby. So to start this, it says parent in group 1 is an AB, which means that is its genotype or a type O. Parents 2, we don't know if they're AA or AO. So this parent could be AA or AO. We don't know. The group 3 parents, they are either big A or big AO. And then the other parent is a B parent. So we don't know what genotype, if they're homozygous or heterozygous for this. However, we got to do the same thing with the babies. So an AB baby is exactly that. A baby who's got type B blood could be homozygous or heterozygous, and a O baby is just got one genotype. So very specifically, we'll start down here. Could parents 3 make an AB baby? Could they get an A from one of the parents and a B from the other parent? And the answer is absolutely. So they could totally make baby 1. What about baby 2? Could they make a B baby? If they took an O from one parent and a B from the other parent, they could totally make a B baby. So this is not super helpful yet. And parent 3s, could they make an O baby? If you look at their genotypes, if they were both heterozygous, they could totally make an O baby. So really, parent pair 3 could make any of these babies. So that's not very helpful. So let's go look at pair 2. Pair 2 have A's and O's, so that means they could make an A baby. But we don't have an A baby over here at the hospital that doesn't know its parents. So that's not an option. Could they make an AB baby? No, they don't have a B in there. But could they make an O baby? Boom and boom. They could totally make an O baby. So it looks like that's their kid. But let's be sure that nobody else can make it, because so far, baby 3 could be made by parents in group 2 and parents in group 3. Let's go take a look at parents in the first group. They have an AB presentation and an O presentation. So that means that you have to take one allele from each of those parents. Could they make an AB baby? No chance. So we can't take both alleles from one parent and nothing from the other parent. That's not how this works. So they can't make an AB baby. Could they make a B baby? If we take this B and this O, absolutely, they could make this baby. So we could put an arrow down here. And then could they make an O baby? Does each parent have one O? And the answer is no. So they can't make this pairing. So what has to happen is one baby's got to go home with one couple. Parents 1 have to take home the only baby they can make, which is baby 2. Parents 2, so we're going to get rid of this arrow. That's not possible anymore because baby 2 is spoken for. Then parents in group 2, they can make this baby 3, this O baby, and that's the only baby of these 3 options that they can make. So that's going to be their baby. So baby 3, and we're going to get rid of this line because that's no longer an available baby. And parent in group 3, they could make all 3 of these babies, but baby 2 and baby 3 have already found their parents. So their baby is baby 1. That is going to be baby AB. Done. So we have all of our babies matched up properly. I think that you can answer these questions. One of those is going to be complete dominance, one of them is going to be codominance, and one of them will be incomplete dominance. So you'll have to figure that out. And then you can see the answers in your notebook. Then I'd like to also go over this one. This was a really important page for sex-linked traits, and they were talking about how this is an example of why we don't want you to keep it in your family. We used to, in early civilizations, we used to try and marry cousins or aunts and uncles because we were trying to keep our wealth isolated in one family. However, what presented again and again and again was this type of presentation where we had what seemed to be perfectly healthy people, and they had perfectly healthy people with an occasional problem, which didn't seem to be too remarkable. But then as you tracked that occasional problem, it got more and more and more common in the family. And the reason for this is because family members are not bringing in new members to the family, and they're not diluting this disease, this recessive carried trait. So if you take a look, the first thing it said was to place XXs and XYs on everybody. And then you had to put the normal allele, a capital D, on all of the individuals who were either perfectly untouched by this blindness, or if they were carriers of this blindness. So they had to put a big D for the normal dominant trait and a little d for the recessive diseased trait. So we'll take a look. There's 4 generations here. I'm not going to talk you through this whole thing, but I do want you to notice the genotype for a carrier woman. We've got a normal male here. Down here, we have a diseased male. There's no such thing as a carrier in sex-linked or a carrier male in sex-linked traits because males have one X and one Y. There's no possibility for them to carry 2 different alleles because they only have one X. And then as you go down, you'll notice in generation 3, we've got a whole bunch of boys who have the disorder, and all the girls here have the disorder, and you have to track who are their parents. So come up to the side. This is someone who married into the family, and they had they had 5 kids, and all their daughters had were carriers. And so we want to make sure we understand why the daughters are all carriers, and you got to pay attention to what dad's got and what dad gives his daughters versus what dad gives his sons. And then pay attention to what mom's got. It looks like she's got a nice protective, dominant, healthy allele, and she gave that to every one of her kids, males and females. But dad gave his Y to his son and gave his X to his girls, which resulted in all of his girls having at least one little d, and that would make you a carrier. However, in the 4th generation is really where it all kind of breaks down. You see this very uneven distribution. So generation 2, there's boys that have the disorder, 2 boys. Generation 3, you've got 3 boys. So it's clearly more males are affected than females. But in the 4th generation, you got every single daughter with this disease. So let's go take a look at their parents. These 2 parents are cousins. So if you take a look at dad's aunt, dad's mom is his wife's aunt. So if you take a look at this lady, who is his wife, her parents are here. This is her father, and this is her father's sister. So your father's sister is your aunt. And so these 2 are first level cousins. And this individual has the blindness disease, and this individual is a carrier. So really, the only ones that have a chance of being okay are, for the most part, I guess girls also had a chance. There's a possibility that they could have had a female who was a carrier. So she would have been a X big D from mom and an X little d from dad. And so she would have been half filled in. But that's not who they had. They had 4 girls in a row who got the recessive allele from dad and the recessive allele from their carrier mom. So that leads me to ask you a couple of questions. So first, do you know that people 1-1 and 1-2 had 8 kids? So people 1-1, that means generation 1, person 1, and generation 1, person 2. They had 8 kids. They didn't have 11 kids. 1, 2 people, or I guess 10 kids. 2 people married into this family. So this person's not a family member, and this person's not a family member. They actually married into this family. And so did this one. So there is 3 non-family members. And what's the relationship between 3-4, so 3-4, and 2-10? So 2-10 is right here. What's their relationship? right? I want to see how they're connected. And then what's the relationship between person 3-5? So 3-5 is this lady, and 2-6, 2-6 is this lady. What's that relationship like? And I'm hoping that you're saying uncle and nephew versus auntie and niece. Then we did, depending upon the class period, we had a little bit more information shared, or some classes had a little bit more time for some practice. And so we went over Mendel's pea plant traits, height, pod shape, and then flower color. And so we practiced. I asked students to come up with a monohybrid cross, which mono means one. So just one of these traits, cross them. So my example might be different than the student examples, but we should make sure that we got an example in here. I took 2 heterozygotes for height, crossed them, and came up with their genotypic ratio and their phenotypic ratio. Again, you could do the short form, 1:2:1, or 3:1 on your test. That's okay. It's just a little risky if you're not confident with what you're doing. And then I asked students to cross 2 plants, just like 2 plants up here. But this time I wanted you to track 2 different traits. So either height and pod shape, or pod shape and flower color, or height and flower color, but some combination of these traits. And so I chose pod shape and flower color. So this is one plant that is dominant for pod shape and dominant for flower color. So this is going to be a smooth purple flowering plant. This plant was also smooth potted, and it had white flowers. So the first thing you've got to do is pick this plant or this, whichever one, and you're going to create all their gametes. This is when we foil. We do the first letter in each pair. Then we do the outside one in each pair, big, little. Then we do the 2 insides, little n, big P. And then we do the last one from each pair, little, little. And those are the gametes. And then over here, we got to get all the combinations for gametes also. So again, we take the first one in each combination, big N, little p. Then the outside ones, so big N, little p. Again, it's a repeat. The inside, again, is a repeat. And then the last one in each of the pairs. And this one was very convenient. I tried to make this as simple as possible. Every single one of these was the same. I think we flipped it in 4th period. 4th period might have been flipped. But then we had to put all of these gametes together. So this gamete fertilized this gamete. This could be the offspring. And that is a smooth potted purple flowering plant. And if, let's see, this sex cell was fertilized by this sex cell, we'd also have a smooth potted white flowering plant. So these are 2 separate, totally separate traits, and we are tracking them together just to see what the probability is of those 2 traits of interest.